Okay, good afternoon. Good afternoon, everyone. I think we'll go ahead and get started. Thank you all for joining us today. It's Tuesday, April 14th. We're at the St. Anthony Falls Lab, and my name is Dan Griffin. I'm a faculty affiliate here at SAFL. My tenure home is in the geography department, and I'm just so very happy today to have the privilege to introduce to you all Professor Mukund Rao. Professor Rao holds a bachelor's degree in chemical engineering and holds master's and doctoral degrees from Columbia University in earth and climate science. After Mukund finished up his PhD in 2020 at Columbia, he accepted the very prestigious NOAA Climate and Global Change postdoctoral fellowship, and which has been held by other folks who work here at SAFL, including Shua Feng and Talia Anderson and myself. He undertook work at the University of California, Davis, evaluating carbon and climate futures with really cool instrumentation. He's going to talk to us a little bit about that work today. Mukund also held a Marie Curie postdoctoral fellowship in Barcelona, and so he is a man of the world, and he spends a lot of time thinking about forests and climate and carbon and instrumentation. And when I had the chance to think about who we might want to invite, I was thinking so much about the opportunity for Mukund to come here. He's also done some really amazing work with statistical modeling and estimations of uncertainty, and we're hoping to wrangle him in to some of the projects that are happening in my group at the time. So today, I think Mukund is going to speak to us at length about work that has been under review at Science Advances for some time now, and I'm so very excited to hear him talk about it. I've had a chance to read it, and so please join me in welcoming Professor Mukund Rao.
Thank you. Thank you, Dan, for almost embarrassingly lovely introduction. And I'm really honored to be here and excited to be here. Are we good on Zoom? Perfect. Okay, so a lot of my research, I think about the forest carbon cycle and how that's changing. And for today's talk, I'm going to start from how trees take up carbon through the process of photosynthesis or assimilation, and then allocate that to tree growth. I am a dendrochronologist or a tree ring scientist, so I think a lot about growth. And the specific, about 80% of the talk, as Dan mentioned, it's this paper that we've had in review for a while about how aridity can decouple the process of carbon uptake and its allocation to tree growth. And of course, this work would not have been possible without help from co-authors. Used a bunch of Dan's Blue Oak data in California, so thank you. And yeah, so let's get right into it, but let's start big about why should we care about trees and forests in the carbon cycle? The annual rate of increase of carbon dioxide in the atmosphere is about 5 gigatons of carbon per year. That's about 2.5 parts per million. But in fact, we're emitting a bit more than double that, closer to about 11 gigatons of carbon. About 90% of that is through the burning of fossil fuels, and about 10% of it comes from land use, land cover change. So we're emitting 11 gigatons, and only about 45% or so of that stays in the atmosphere. And the reason for that is we have these natural climate sinks that are in many ways mitigating us or protecting us from some of the worst impacts of climate change. About a quarter of carbon that we emit ends up in the ocean, and then about a third of that ends up in terrestrial ecosystems, forests playing a large, important role, but other ecosystems like wetlands are also incredibly important. Importantly, I focus on the terrestrial ecosystem sides of things, and they also, on an annual basis, take up and store a lot of carbon, but they also have a large stock of carbon. So if we were to do a simple mathematical exercise of where would we be if these natural carbon sinks were not functioning back in 1958 when we started measuring carbon dioxide concentrations in the atmosphere at Mauna Loa. We were at about 315 parts per million. And then right now we're at about 425, 426. And prior to the start of the Industrial Revolution, we were closer to 270 or so parts per million. So if every year we are emitting, let's say 10, in the present, 10 or 11 gigatons of carbon, and 60% of that goes into the land and the ocean, we could calculate where we would be in the present if these sinks were not working. So I ran this out, I think, to 2024 or so. So then we were at 420 parts per million with an increase of 115. But if these natural carbon sinks were not working, we would actually have been at 550 or even a bit higher than that. So in many ways, actually, the experiment that we're doing on the planet, we've already doubled. We would have doubled carbon dioxide concentrations, but our ecosystems and the ocean are protecting us. So there's a large concern that the capacity of forests to continue to mitigate these or take up carbon and might be increasingly under threat due to heat waves and drought. And so some of the motivating factors behind my research, and just figure out why this offset happens and how it might change in the future. So let's think from the perspective of a tree. What are the main important processes? Trees are taking up carbon dioxide through photosynthesis, and in exchange also transpiring water. And forests are open systems. There's only one way for carbon to enter the ecosystem, and that's through photosynthesis. But there's many ways out for that carbon. So the carbon might be a large fraction, or more than half of that carbon is respired out directly by the plants. Some of it enters the soil and then is respired out through decomposition. And some fraction of the carbon is allocated to both above ground growth, below ground growth, and other biomass such as plant foliage. And there's also other processes that can store carbon at longer timescales, but a small fraction of what's taken in. Importantly, I focus a lot on the processes of photosynthesis and tree growth. The environment plays a really important role in regulating these processes. And then photosynthesis and tree growth are actually under independent environmental controls. So traditionally, if we think about how many Earth system models are formulated, it's you have photosynthesis, and then growth, respiration, and storage are all just some fraction of these processes. And if you have photosynthesis, you always have growth, but we'll get somewhat into the details of these. You're going to say something? So why growth? Why should we care about growth for carbon storage? That's because woody biomass can stay on the landscape for centuries to even millennia in some contexts. These are sequoia coastal redwood trees, Brisicone pine trees, Fitzroya cupressides in Chile. Those are some of the exemplars of long-lived trees, but even out here, oak trees and spruce trees can live for hundreds of years at least, maybe longer. And so we're going to start with two big questions for today. The first question that we're going to look at we're going to compare the phenology of growth and photosynthesis. And the second question is we're going to look at the environmental sensitivities of both processes. So photosynthesis, or carbon assimilation, that's how carbon went in, and growth is how it might stay on the landscape for long-term scales. So on the photosynthesis side of things, we're going to use a bunch of ways to estimate this. We're going to use digital repeat photography using phenological cameras, or phenocams. We're going to use satellites, and specifically we're going to use an index known as solar-induced chlorophyll fluorescence, and that's a small red glow that plants emit when they're actively conducting photosynthesis. We're going to use flux towers. Flux towers measure or look at atmosphere gas exchange of carbon dioxide and other gas exchange of carbon dioxide between the atmosphere and biosphere as you have turbulent eddies moving across the canopy. And then we also made other measurements like leaf level chlorophorescence. And in the growth side of things, we're going to use a network of point dendrometers. Point dendrometers are, and we had some discussions around this in the morning with colleagues, where these are sensors that you can put on top of trees. There's a spring over here, and as the tree is growing or expanding, it displaces the spring. So in daily timescales, you can see things like how the tree shrinks during the day as it's transpiring water, expands at night as it's taking up water, and then during the course of the season, you can see how trees are growing during the entire season. We use quantitative wood anatomy, which we take small cores from trees to look at the cellular scale, when are they dividing. And I'm going to talk about tree rings, but not in this part of the talk, but there's a lot of tree ring data as well, and I'm happy to share the paper if anyone's interested. So we're going to look at 4 sites across the US, and we're going to focus on deciduous oak species at all of these 4 sites. The closest site to here is a site in Illinois. That's also our coldest site. We have 3 sites in the eastern US and a 4th site in California. The 3 sites in the eastern US, they have more or less even amounts of precipitation during the entire year, and you get a bit warmer as you go from the 1st to the 3rd site. And then the 4th site in California, that's a Mediterranean oak forest, still deciduous, but rainfall mostly falls during winter and spring. Summers are hot and dry. And then the little symbols here tell us what kind of measurements we're making at this site. So camera, satellite, derived GPP, point dendrometers, and then flux towers at 2 sites. And we do this for 7 different oak species at each of these sites. So just because vapor pressure deficit, or how dry the atmosphere, is going to play an important role in this talk, let's look at the climatology of vapor pressure deficit across the 4 sites just for 1 year to make the comparisons easy. Here we have January, December. This is smooth with a 15-day average. And this site in Illinois has a really cold winter, warm summer, while the site in California has a cool-ish winter and very hot and dry summer, so high vapor pressure deficits during the summer. And over here I have some statistics. And the coefficient of variation, the CV, is the coefficient of variation of vapor pressure deficit during the course of the year. So how variable is it during the course of the year, let's say between winter and summer?
Excuse me, sorry.
Yeah.
What is that?
So
What's vapor pressure?
Vapor pressure deficit, it's a metric of how dry the atmosphere is. So how much water would it need to become fully saturated? So it's a function of temperature and humidity. As the atmosphere warms, it can hold more water vapor. So as it warms, you need it's the atmospheric demand for water. Very good question. Yeah, Kimberly.
Somehow I thought that it was also having to do with the moisture that's inside the tree or
Yes.
something like that, or no? Is it just, yes? Usually
Yes, it does. So we are thinking from the perspective of the tree. If a tree is fully saturated and the atmosphere is very dry, it's how quickly would the atmosphere be demanding or taking away water from the tree. So it's the gradient that's actually really important. Thank you. Yeah, and I think after discussions this morning, realized we have a very diverse audience working from fluid dynamics at various scales to thinking about experts on tree rings. So please feel free to stop me and ask questions. Totally okay with that yeah. So let's start big picture and compare the phenology of growth versus photosynthesis. And we're going to start with the site in Illinois, which also may be climatologically similar to here. We have 3 different oak species that we are measuring at the Morton Arboretum. close to Chicago. And I have 3 different panels here. This is the green chromatic coordinate that we get from the phenocam. So we're taking pictures every 15 minutes of the trees and then looking at an index of greenness. And then these are data for 2021, and the x-axis here is time starting in January through December. So here, our GCC time series is kind of what we'd expect for a typical broadleaf deciduous forest. Not much happening in winter. In spring, around now in mid to late April, the leaves begin to come out. The canopy greens up. We reach a peak of greenness in mid-spring, late spring, and by the time we reach May or June, the canopy remains green till about end of mid to end late October, and then the canopy senesces during the autumn. Then we have GPP, or gross primary productivity, and in this particular case, we get this from satellite measurements of chlorophyll-fluorescence. So this red glow that's closely tied to photosynthesis, and we see that GPP tracks canopy greenness pretty closely. Once the canopy greens up, or when you have no leaves, you cannot be plants are not photosynthesizing. Yes. And then once the leaves come out, they are taking up carbon dioxide, and the fastest rates of this happen during spring to early summer. And then when the canopy senesces, the GPP goes down as well. And finally, the third panel here, the lowest panel, is the fraction of annual growth, and we're going from 0 to 1. And this we get from 18 trees where we have these dendrometers in this arboretum. So, and we've standardized them from 0 to 1 just to standardize across all the trees. And what we see is that the trees begin to start growing in late April or so, and then this is their phenology of here we see the phenology of their growth, which is quite a bit different from the phenology of GPP and canopy greenness. So the trees begin to start growing in late April, early May, and then by the time we reach mid-August or so, they're done growing or expanding for the season. So then we divided this annual season or productive season into 3 different phases. The first phase, over here, the canopy is beginning to green up, gross primary productivity is increasing, and the trees have already grown about 40 to 50% of their annual growth even before the canopy has fully expanded and greened up. Then there's a second phase where the canopy has fully greened up, the leaves have expanded, and the trees continue to grow till about mid-August or so. And then there's this third really long phase where the trees continue to have green leaves. The forest is still taking up carbon dioxide, albeit at a slightly lower rate than earlier on in the spring and summer, but the trees are done expanding or growing for this season. And so if we look at these 3 different phases, in this first phase, we have active growth and carbon assimilation, or APE, is beginning to ramp up for the season. In the second phase, both processes are happening together. And then in the third phase, growth is done or down-regulated for the season while carbon assimilation continues. So then now, just to make it compare across the sites, what I've done is I've combined all of that information just into one plot. So we have 3 different lines here: GCC, or the green chromatic coordinate from the phenological cameras, growth in purple that we get from the dendrometers, and then GPP, or gross primary productivity. At the first 2 sites, we get that from satellite remote sensing, and at the third and fourth site, we get that from in situ measurements, and then we can look at how these phases vary across the different sites. And we calculated this index known as an offset, which is how big this second phase, where GPP and growth are happening together, relative to the entire first phase, where you have growth but downregulated carbon assimilation, and this third phase where growth is done but assimilation continues. So just to make it easier, the bigger the offset fraction, the smaller is the second phase and the bigger are the first and third phases. So it's like some kind of metric of the coupling of growth and photosynthesis across the season. And we did this for different sites and we get different numbers, but the broad pattern is similar. And for example, at this California site, we see that growth and photosynthesis peak really early, but then there's a long phase between about most June through October or so where the trees are not expanding anymore or they're done growing, but the forest continues to take up carbon dioxide. So in this case, this is actually the site where carbon assimilation or photosynthesis and growth are most offset from each other. So why does this happen? We wanted to then investigate the environmental sensitivity of carbon assimilation in comparison to tree growth. So let's go to the site in California. We have the same three different phases, three different data streams, the green chromatic coordinate from the phenochem, gross primary productivity, and growth fraction with dendrometers. The only difference here is that we get gross primary productivity at this site from a flux tower that we have on the landscape. So it's at the same time resolution as the dendrometer. It's measuring gas exchange between the atmosphere and the biosphere, so we can then make an apples-to-apples comparison of when is the forest taking up carbon dioxide or photosynthesizing. So when do you have gross primary productivity relative to when do you have growth that you get from the dendrometers. And similarly, we divided this active season into these three different phases. And for now, we're just going to focus on the second phase when we do have both growth and GPP are happening together to make the most fair comparison of what is the environmental niche for GPP or carbon assimilation relative to tree growth. So that's the plot here. X-axis is air temperature, y-axis is vapor pressure deficit. That's the metric of how much water is the atmosphere demanding from the plants. So greater vapor pressure deficit means that the atmosphere is very dry. And it also there's an exponential relationship between air temperature and vapor pressure deficit, with the additional variable being humidity that influences vapor pressure deficit. And the green shading here and the size of the circles is gross primary productivity. The darker the green and bigger the circle, more amount of GPP, so more photosynthesis. So what we see is that photosynthesis generally peaks around, let's say, 10 to 18 degrees centigrade and about 0.5 to about 1, or 0.25 to about 1 kilopascals of atmospheric vapor pressure deficit. So onto the same plot now, I've added a bunch of purple points, and this is what information that we get from the point dendrometers that tell us where does growth happen within the same environmental niche. So we're comparing at the same half-hourly time step, when do you have growth versus when do you have photosynthesis. And what we see is that growth, or the allocation of carbon to biomass, is happening at significantly cooler air temperatures and much lower vapor pressure deficits. And this makes physiologically a lot of sense, because when cellular division at the tree scale needs water for cellular expansion. And so when you have warm temperatures and the atmosphere is demanding a lot of water, the trees and the leaves are still able to transpire water and assimilate carbon dioxide, but at the cellular levels, when cells are dividing, they stop growing, but they can still take up carbon dioxide. So then we wanted to look at, because vapor pressure deficit seemed to be a really important variable at one site in terms of decoupling carbon dioxide assimilation from growth, we wanted to see if maybe there's a relationship between vapor pressure deficit and how much decoupling between photosynthesis or the offset variable across sites. And what we found is that there's a pretty strong linear relationship between the annual coefficient of variation of vapor pressure deficit and offset across all of the 7 sites, all of the 4 sites and 7 site years that we had. Of course, this is a pretty limited data set, but what we seem to find indication for is that as vapor pressure deficit becomes more variable during the year, where the greater the amount of offset or decoupling between photosynthesis and growth. So make it easy. If at a site you have relatively similar water availability and temperature throughout the year, maybe that favors plants to always be taking up carbon dioxide and growing at the same time. So maybe in a tropical forest, photosynthesis and growth could happen at the same time, while in a forest that maybe has a very strong wet season and a very strong dry season, a very cold winters, or short and warm summers, where climate becomes more variable, maybe these processes begin to start becoming more offset from each other. So this is at the inter-annual scale, but then we also wanted to look at the daily scale to see if there's any physiological mechanisms of why photosynthesis and growth might be decoupled. So the x-axis here is the hour of day. So we start at midnight and go to 11 p.m. or 23 hours. And I'm going to start with we're going to have 3 different curves here. In purple, we have growth that we get from the point dendrometers. The green is going to be gross primary productivity or photosynthesis. And both growth and GPP are going to sum up to 1, because we are averaging growth and photosynthesis across the entire growing season, the active season. And then we're going to add in another variable called tree water deficit. So tree water deficit, using the point dendrometer data, you can separate it out into 2 phases. One is each time the dendrometer reaches a new maximum, that's growth. But you also have this daily cycle of expansion and shrinkage, and that's known as tree water deficit. So as the trees begin to shrink more and more, that means that the tree is it's shrinking because it's losing water, and if the tree is expanding, that's because it's rehydrating. So each time tree water deficit is negative, it's actually in excess. You can think of well-hydrated tree, and then if tree water deficit is positive, this means that the tree is shrinking because of water stress. So let's start from 0, so midnight to about 7 in the morning, and this is what we see for the trajectories of growth, GPP, and tree water deficit. So during this phase, we can see that as we go from midnight to about 7 or 8 in the morning, the fraction of annual growth is increasing. So almost 80% of growth actually happens during this phase from late night to early morning. And then as we reach about 5 or 6 in the morning, as the sun comes out, once you have light availability, that's when the plants are beginning to photosynthesize. And if we look at tree water deficit, it's becoming more negative. And this is because the trees are refilling their water reserves through their roots during the course of the night, and at this phase, they're not losing water due to transpiration at the leaf scale. Once the leaves come out, this is still early morning to about midday or so. The moment the sun comes out, what we see is gross primary productivity or photosynthesis continues to increase. That's because for photosynthesis, you need light. But the moment that happens, the fraction of growth decreases actually really rapidly. And this is because once the sun comes out, the plants are transpiring water, and in the cambium, or where the trees are growing in their trunks, this water stress inhibits cellular division. And so the trees, once the sun comes out and the trees are taking up carbon dioxide, they're unable to actually grow because at the cellular scale in their tree trunks, the cells have the turgor pressure in their cells is lower. And during this phase, we can see the tree water deficit begins to start becoming more positive. And that's because the trees, earlier on in the morning, they were expanding because they were taking up water, and now they begin to start shrinking. And then the second half of the day, I have to wait a few seconds. Okay. And finally, after midday, this is when the sun is more intense, gross primary productivity continues, but it decreases as we reach into the evening. And we can see that the trees are almost not or never grow during the second half of the day between about midday to midnight. And tree water deficit continues to increase till about 5 or 6 in the afternoon. This is because during the second half of the day, the sun is still pretty intense and the trees are continuing to transpire water, and they're under more increasingly positive tree water deficit, means they're more water stressed during the second half of the day. And all of this begins to relax only as we reach, as the sun begins to set in the evening after about 5 in the evening, where tree water deficit begins to relax as the sun sets, conditions are less intense, so the tree can begin to rest and recover and take up water again. Do you have a question?
I do. Thank you. This is fascinating. I'm wondering how to think about this picture in regards relative to the changes across the summer, right? Is this a typical spring day, or is this on average, or?
This is on average, but actually I don't have graphs here, but you can see even how as you move from spring into summer, this timing of peak growth shifts earlier and earlier because the sun comes out earlier and earlier, and then it relaxes back to being later as we pass the solstice. So this is averaged across the entire productive season, but it changes a little bit. Things move around.
But isn't it also true that maybe like in August, it sums to zero, right? So there's some positive and some negative as the tree takes up water and then loses water at the end of the day? Is that like a form of
Yes,
growth?
I think so. And then if you have a heat wave or the trees, you go many days without any water, also maybe the trees that might not even grow at all during the night because they're so water stressed. So this is big picture across the entire season, but how it varies within the season is a pretty interesting question and things that I'm very interested in.
Thank you.
I don't know. Okay, so we were talking using dendrometers, and dendrometers are measuring tree growth at kind of outside the tree scale, and we were interested in verifying to make sure that what we see at the dendrometer scale actually matches up with what's happening at the cellular scale. So we collected these microcores from trees every couple of weeks. This is not an oak, but we did this at all the oak trees on one of the sites to really see at the cellular scale how does the ring form. And so this is the site in New York when we collected microcores every 2 weeks, more or less, in the 2021 calendar year. So oaks have this really interesting wood anatomy, and they're known as ring porous species. And if you look at the annual tree ring of an oak, the early wood of the oak has these really big vessels and that allow it to transport a lot of water. And then as you reach into the second half of this oak tree ring, or the late wood has much smaller vessels. And so by collecting microcores, we were able to figure out how exactly the hydraulic conductivity of the tree ring changes during the course of the year. So let's start in April 1st. This is before the dendrometers have begun to show any growth. We found only evidence for the 2020 growth ring. Then in April 19th, we saw that the trees have begun to form one row of vessels in the month of April, so they're starting to form their early wood. And by the time we reach May and June, most of the early wood of the oaks are formed, and then by June and July, they've already switched to forming the late wood of the oaks. And then by August and September, the trees have already almost finished all of their annual growth for the season. And this is very consistent with the dendrometer data that showed that by the time we reach the month of August in New York, the trees are done growing. While they maintain their leaves on the canopy and continue to take up carbon dioxide through all the month of September and even October into late October, early November. So there's, even at the cellular scale, we can think of these offsets. And so to conclude this part of the study, while you can enjoy a GIF of a year in the life of a forest, in the spring, we found that tree growth occurs under low photosynthetic capacity. So the trees are growing actively even before the leaves have fully come out for the season. And in the autumn, carbon assimilation continues without growth. So these two processes are decoupled from each other, the annual scale. And growth occurs both at lower vapor pressure deficit and temperature than carbon assimilation now, both at DL or daily and at seasonal scales. And so most Earth ecosystem and Earth system models, the way that they're programmed up is very photosynthesis centric. So if you have photosynthesis, you always have growth, and you have the carbon budget. Most models try to balance out the carbon budget of a tree or an ecosystem at every time step. And what we're finding here is that's really not the case, where the trees might be growing, might be continuing to take up carbon in the autumn, and they probably use those reserves to start growth next spring before the leaves have fully come out. So there's a lot of maybe much more dynamicism to carbon uptake, storage, assimilation, and mobilization that we need to start thinking about. And finally, the aridity of the atmosphere increases decoupling both now and likely into the future, because one of the main impacts of climate change is the increase in vapor pressure deficit, even more so than temperature, because vapor pressure deficit increases exponentially with temperature. So some things that I'm working on right now is I'm putting together so this is only 4 sites in the US, but we're putting together a large network of sites where we have GPP or photosynthesis, flux tower measurements, and dendrometer measurements at the same site, and then trying to look at these processes across a larger environmental ecosystem gradient. So if any interest in collaboration, I would definitely be happy to chat. And in the last 5 to 10 minutes, I want to talk about a couple of other research directions that we are actively working on in our lab group. And so for the first part of the talk was mostly focused on drought and water dynamics, but there's another really important variable that can impact forest carbon cycling, and that's extreme heat. The left-hand side is an image of forest mortality in Mongolia, in the southern edge of the boreal forest. And this is a region which is warming about 3 to 4 times faster than the global average. And the increasing heat events are, when you have a heat event, and especially if the heat event is coupled with drought, it can raise leaf temperatures, and the temperature at which leaves die. At some temperatures, the leaves begin to sustain damage and potentially die. And so during heat events that are both the frequency and magnitude of these events is increasing, there's increasing concern that we might be reaching tipping points where after heat waves, forests might not recover. So in a study that we have under review right now, we've collated these in the field we went out, warmed up leaves, and then figured out at what temperatures do these leaves die, and then used Earth system models to figure out how close are the leaves to their thermal tolerance maximum. So we calculated this index known as the thermal safety margin. So that's what temperatures do the leaf die at, and where are air temperatures right now, and where will they be in the future. So this is just using data from 2001 to 2020. And any region in red over here, which is most of the tropical forests, especially in southeastern Amazonia, what we see is that the leaves are getting, as the air temperatures are warming, the leaves are getting closer and closer to their thermal tolerance thresholds, which is concerning. And so thinking big picture, most Earth system models project that as we move into the future, as carbon dioxide concentrations increase in the atmosphere, plant food, so-called plant food, that would increase ecosystem productivity globally, regardless of which scenario we follow. And so you have this lush green world, but there's a whole range of reasons why maybe most our Earth system models are overly optimistic. Fire, insect outbreaks, heat waves, drought, diseases. So, and many of these processes are actually not very well understood and not very well accounted for in Earth system models. So there's actually a lot of concern that the reality might not be this optimistic, and we probably might be lower down on this range. So this is some of the things that I'm working towards resolving. And last bit of thing, kind of other line of research is looking at archaeology, dendroarchaeology. So tree rings, and at least for the tree ring folks in the room who have not yet seen proper tree rings. And all of these ancient structures have a lot of information contained in them about past human society and past timber transport. So in New York City, for example, there's active urban renewal, and along with urban renewal comes demolition. And we're actively working on collecting timbers from these demolition sites. In many cases, these timbers came from historic old growth forests that no longer exist. So this is what forests were in the 1850s, and this is what forests were in the early 20th century. And most of these forests disappeared because they were used for timber use, heating, and a whole range of human uses. But some of those timbers remain preserved to this day in wooden building structures. And we've been trying to figure out these stories of past timber transport. So for example, one of the studies that we worked on were the remains of a ship that we were found off the Argentinian coast. And this ship was known as the Dolphin, was a ship built at a shipyard in Rhode Island using white oak timbers from Massachusetts and longleaf pine timbers from the southeastern US, crashed off the Argentinian coast. It was a whaling ship. And along with colleagues from Argentina, we were able to figure out, once we collected samples, we were able to see that these timbers have North American species, and then using dendrochronology, figure out where the origin source of the timbers and when they were cut. Similarly, we always go hunting for timbers whenever we see them, because this is not landfill garbage, it's actually environmental information. And last slide I have, this is the most recent thing I've been working on. A couple of months ago, someone brought in a painting to our lab, and they thought that it's a painting painted by this Flemish master painter, Peter Bout, who worked in the 17th century. And the side of the painting, you can see these beautiful tree rings on oak. And the oak panel painting style was a really important style in Europe. And after the Renaissance period, most painters switched to the canvas as the medium of painting. But pre-canvas, most paintings were made directly on wooden panels. So here, the cool thing about wooden panels is that they have tree rings, and then you can study the tree rings, measure them to figure out, would this painting would the wood that was on which this painting was painted, could this correspond to a Peter Bout? Because he just bought this at a random auction and once asked us if we could at least validate that the wood material would come from this particular time and region. So that's what the oak panel looks like at the back. There's a whole range of chronologies of tree ring sites across Europe. we measured our treating series and then correlated this with treating series across Europe. This is a Flemish painter, so they'll be working somewhere in this region. And we found that, yes, it correlates reasonably well. Here in black is the treating series of our oak panel. And then in yellow and red are two treating chronologies from France, also oak chronologies. And what we see is that the annual pattern of wide and narrow rings in our oak panel painting match almost perfectly with living trees or the treating chronologies from this side. The last ring on our oak panel was 1622. This painter was known to be active in the 1650s or so. And actually the painter Peter Bout, even though he was Flemish, he worked for many years in Paris. And both of these treating chronologies that we have the best match were French chronology. So could, at least, we cannot tell him that this was painted by his person, his painter, but we can at least say that the timber is consistent with, and the source region is consistent with the panel that he would have used. So just to end on more different, interesting note, happy to take any questions, and thank you for your attention and the invite.
Thank you so much. We do have time for questions, and I'll bring this mic around if someone has one. Hi.
Thanks.
So there's a point that I thought I understood early on, and now I realize I'm not sure if I understand when you showed three things that you were measuring in these different oak forests and oak species. And I feel like one was the rate of photosynthesis. And
This one? Or
even in the annual one, maybe this one, but even before. But you measured photosynthesis and you measured growth, and mostly it's growth outwards. Is that the idea? Okay. And then what's the third thing?
so here, this is leaf or leaf foliar phenology. So when do the leaves come out, when are they green, and when do they senesce?
Okay, so that's kind of like a visual reading of greenness. And then how do you measure the photosynthesis reactions? Can you repeat that?
Great question. So we have these towers, and what the towers are doing is that they have sensors that are measuring both wind speed, the direction of the winds, and they're also measuring carbon dioxide concentration. So if you have wind blowing across the top of a forest, it creates eddies, and then these sensors are fixed in space. So if you know the direction of the wind, if it's going, let's say the wind is going down into the forest, and you're measuring carbon dioxide concentrations, and then after the eddy passes, then wind is returning back, you can say the direction of carbon going from the forest to the atmosphere. And so at each time you're measuring this directionality of the carbon exchange, and when the forest is taking up carbon dioxide, so during the day, and a good day, there's sunlight, the forest is taking up carbon dioxide, what you'd see is in the gradient going down versus going up, you would see that the forest is taking up carbon dioxide, while at night, the forest is releasing carbon dioxide from the biosphere to the atmosphere. And then because we are measuring this continuously in time, you can look at a biosphere or ecosystem scale, so it would be a few square kilometers, how is the forest exchanging carbon with the atmosphere?
So is it sort of like using eddy, sort of our knowledge of, eddy, so like correlation,
theory.
Wow, okay. And so the significant, in some ways, part of the significance, I think I'm getting this partly from you explaining this morning, is that we might think of photosynthesis when that's happening most actively. We also have it growing most actively. And this is showing no. And on top of it, we might guess it, right? I think, okay, I've got to leave the leaves on my irises longer than there are blooms so that the plant can absorb more energy. But this is actually can be a measure of that, what energy is creating to absorb.
Yes.
Is that right? Okay.
That's exactly, and I think thank you, Kimberly, that's exactly where I was getting at. And even conception or folklore about when do you water your garden, when do you water your plants, why would you want, if you water your plants at night, that's considered the best because that's when the plants are growing new leaves or doing things with that water. If you water them in the day, they might just lose a lot of that water. And so that's like, when would you irrigate your crops? When would you water your house plants? Generally always think of watering plants at night. And so it makes even like that.
And
the
that's
irises-
consistent with your measures, right?
Yes.
Yeah, okay, very, very nice, thank you.
Thank you. That was a fascinating presentation. I just have a short question, which is, if you were to replant that blue oak in New York, would you see the same behavior? Is it like encoded in the tree, or is it in the climate?
That is the nature versus nurture question, and it's a hard question. I don't think, but New York, in many ways, yes, this is an extreme system, but the blue oaks are incredibly adapted to that. We have the blue oak expert in the room here, so we should maybe ask them, but I think they're incredibly well adapted to this more Mediterranean, dry, hot landscape. If you give them, I think the issue would be maybe it might do well, but it's the other trees in that region that are well adapted might do even better. So they would just never be able to compete with other trees if you were to take it to New York, for example. But in that environment, in California, where it's very hot and dry in the summer, it does much better than any other tree species around. So I think it's uniquely suited to its niche. Do you have anything you want to add?
Sure. So I would add that Blair McLaughlin, who I think is at UC Santa Cruz lately, she has been doing these common garden experiments where they bring seed stock for blue oak from different parts of its climatic envelope, plant them in a wetter place that might be the kind of thing where you would target assisted migration. And ultimately the question here is, is the fate of these trees genetically controlled based on the kind of, biogeographic origin, or is it a result of the environmental conditions in which the tree germinates and experiences the early phases of the life course? And those are the kind of, my understanding is those are the bifurcating theories, and it's real tricky. I think what I heard her present recently was the latter. It's almost more of what's happening when the tree is young, and maybe it's less genetically controlled. But one of the things, if I could extend this question into my own, I wonder about your mechanistic diagram. You've got the 3 panels. Can you go forward like 5 or 6 slides? Because I wonder how these things vary across the life course. And we're talking about organisms that might live for centuries. What's going on when these trees are, young kind of whippersnappers versus old curmudgeons? We got all flavors in the room here. So, how does this phenomenon vary as a function of where trees are in their social status or in their aging process? Do we know?
That's a really good question. So our sampling strategy at most sites this far has been partially due to financial limitations, putting dendrometers at about 5 to 10 trees. But if we get help to build cheaper sensors, maybe we can instrument all trees across age classes, all species, then we can answer them.
Sorry.
Would be interesting to do that.
Question. Another question.
Oh, I was just gonna chime in on the nature versus nurture. Janine Cavenger-Bears has a bur oak experiment where one's at Cedar Creek here, one's at Morton, and one is somewhere more southern. And it's nature and nurture. So where they came from depends. So they took seeds of origin of bur oak from these 3 sites and did reciprocal transplant experiment. And depending on where they came from, the trees will grow differently. So it's nature and nurture.
Thanks,
I think,
Professor Smith.
to me, that makes sense, that it's a bit of both.
But it's site of origin and where they grow.
Okay.
So if the tree is producing carbon, which I think you said for a significant period when it's not growing, where's the carbon going?
I think a lot of that is being stored temporarily
You
as
sort of indicated
non-structural.
that in the conclusions, but what, I don't understand that. Like, where would it be stored and?
It can be stored in many places. It can be stored in living tissues, in oaks, in fibers, in the roots and stems, and then be remobilized for growing or all other plant processes the following year, or it can actually be stored for many, many years. So they're known as non-structural carbohydrates. They're a bit tricky to measure, but maybe Chris wants to measure them. I think she wants to, so.
But this is how they're growing before they even have leaves. They have to have stored food. If not, how are they growing before they've even leafed out?
Yes. But getting into the anatomy of the oaks, I find oaks very fascinating, because if we go to the previous year vessels of oaks, so that's the previous year, generally they're not very functional. And so to even expand leaves, the oaks need to transport water from the roots to the leaves to expand the leaves. And because the previous year vessels, the transport water are not functional, it makes sense that they grow even before the leaves come out, because this growth is what's helping them move the water up to the leaves and then expand the leaves. So, and they have to be using stored carbohydrates to do that, because this is a pretty carbon intensive process.
Following along with these thoughts, I was wondering when the food is being stored, is there a general belief that we know is there a scientific theory that's been backed up that says where, are the roots getting bigger for a little while? But it happens in August, when I'm leaving my leaves on my irises. And then in the spring, are there actual physical shrinkage of the roots maybe? Or do we not know? Like when we think about that food being used, where is it being used from? Is that, and it's a really simplistic idea that I'm thinking, because I'm thinking, okay, Christmas cookie time, I store a little bit extra, and that's in a place that I'm not going to talk about, but and then a little bit after that, maybe I spend a little extra time at the gym, and then that place gets a little smaller. And so I'm wondering, is there any analogy like that for the trees that is known, or are there just a bunch of essentially hypotheses that haven't really been studied extensively?
Excellent question. I think this is definitely at the forefront of what the community is doing. I think traditionally making these kind of non-structural carbohydrate measurements, you might do it once, but then the way to really get at this is to do them continuously to figure out how dynamic these are. And so most studies we have now, I think one traditional way of doing this is you could say give a plant enriched carbon dioxide, so you can spike the air that plant is taking up with C14, and then see how long does that C14 take to reach the stems, or take to reach the fruits, or take to reach the roots, to figure out something about transit times of carbon within the plant system. But you could only really do that in a lab setting where you grow the plant in a chamber and monitor everything. Doing this at a tree scale becomes much more challenging, but I think people are definitely working, thinking a lot about this. Just the logistics of measuring dynamic processes are harder.
Thank you.
Thank you.
Time for one more question, if there is one, perhaps.
Maybe. Thanks.
Thank you for the talk. It was amazing. I was wondering if you did the anatomy for all of oaks or just the alba, And if you did, like, did you find difference in the anatomy, like, the dimensions of the xylem vessels? I was wondering about the offset that you found, the difference between the growth and the photosynthesis. And I was wondering if there is some relationship between the water transport capacity of the tree with that offset. I don't know if you had any
Yes.
thoughts about that.
Whoop. Okay. I might have crashed out of Zoom. Sorry. But I don't know if, were there folks on Zoom?
No, we're not sharing anymore, but.
It's, well, I do actually have, the metric that we did was relative conductive area. That's how much of the ring was conductive vessels. And then this is as the microcores progressed in time from March until September. And what sees as the trees are beginning to grow in April and May is when you have more than 60% of the ring is just these big vessels transporting water. And then in the second half of the season, you're producing these vessels that are much smaller, so the conductive area decreases. But then there's many other wood anatomical metrics you can figure out. The lumen area and the theoretical water transport capacity, and even this kind of wood anatomy is definitely relatively new in the, at least to figure this out in time. But there's many things we could do with bigger pipe means you can transport more water, but bigger pipe also means there's greater risk of cavitation. So there's all of these trade-offs that plants have to deal with. And I'd be happy to chat with you more after.
Okay, please join me in giving Mukund another round of applause.